Greetings from Team iGEM NCKU Tainan. Hope you have learned a lot from our previous audiobook. There are also 3 topics today. It's In My Genes, COVID-19 detection mask, and Turn to Better Nitrogen, respectively. Now, let's get started on our audiobook episode 2.
It's in my genes. We often say it is in someone's genes when the person has the same personality traits as his or her parents. For now, this proverb is not an exaggeration anymore, since a company called Codex DNA has successfully stored a classic song, Hey Jude from the Beatles, into DNA for data storage. Codex DNA is a company devoted to inventing automatic gene assembly techniques, such as solar and Gibson assembly, to make DNA synthesis faster and cheaper than directly buying from some specific companies. Solar, also known as short oligoligation assembly, uses an universal pre-manufactured library of short DNA building blocks to construct oligo sequences up to 100 base pairs. For applications requiring longer builds such as gene, mRNA, or protein synthesis, the 100 base pairs products can be further processed using well-established methods such as Gibson assembly. Gibson assembly is a molecular cloning method that allows the joining of multiple DNA fragments in a single isothermal reaction. The method can simultaneously combine up to 15 DNA fragments. Based on the sequence identity, the Gibson assembly requires the DNA fragments to contain 20 to 40 base pairs that overlap with adjacent DNA fragments. These DNA fragments are later mixed with a cocktail of 3 enzymes: exonuclease, DNA polymerase, and DNA ligase. Now, we can store the digital data by converting the binary codes of 0s and 1s into the A, C, G, and T, building blocks of nucleic acids. The storage capacity of a DNA molecule is significantly higher than that of standard data storage technology, with the potential of encoding more than 200 petabytes of data per gram of DNA. Moreover, it lasts longer. DNA has been shown to remain intact for more than 1 million years. As a result, this method of storing and retrieving digital data using DNA revolutionized the way how data is stored. Codex DNA researchers used the company's proprietary software algorithm to encode data files into a DNA sequence. The team used this approach for an audio file, a brief snippet from Hey Jude from the Beatles, and also an image file of the company logo.
COVID-19 detection mask. In 2021, Harvard University and MIT have worked together to invent a mask that can detect COVID-19 by sensing the breath of the user. By pressing the button on the mask, users can easily get the result in 90 minutes. Without using living bacteria, the synthetic circuits was applied to the mask successfully with the FDCF technique, followed by the RT-RPA, CRISPR, and LFA systems. The results will be finally shown on the test paper, which can be easily read by the users. What is FDCF? Its full name is the freeze-dried cell-free system, which is composed of a cell-free protein synthesis system and the freeze-drying technique. The former is a rapid method to produce proteins in solutions without involving cells. The latter is the most widespread method to preserve biomaterials by dehydration. RT-RPA, also known as reverse transcription recombinase polymerase amplification, is an isothermal alternative to RT-qPCR. It turns the RNA sequence of coronavirus into cDNAs by reverse transcriptase and then amplifies the DNA sequence to reach enough amount for detection. Lateral flow assays, LFA, is a paper-based platform to detect a liquid target analyte. When the liquid flows through the test line and the control line, the antibodies or aptamers will mark the target particles. It is also the technique behind COVID-19 rapid antigens tests.
Turn to a better nitrogen. Do you know that we can produce fertilizer in a biological way? Crops usually rely on synthetic nitrogen fertilizers, yet the use of chemical fertilizers are neither efficient nor eco-friendly. About half of the nitrogen fertilizer applied to crops is lost in the environment before reaching the plants. The use of synthetic fertilizer can lead to several devastating environmental effects, such as water pollution and ozone depletion. In addition, the production process consumes a staggering 1 to 2% of global energy per year. Biological nitrogen fixation, BNF, is a solution. By remodeling the genome of the Klebsiella varicola 137, KV137, the company Pivot Bio has created the first biological fertilizer for corn, Proven, which can directly adhere to corn roots. KV137 contains the NIF-L and NIF-A genes, which can control the nitrogen fixation pathway. By replacing the inhibitor NIF-L with a constitutive promoter, the NIF-A protein can be produced constitutively. Hence, it can fix nitrogen from the air regardless of the exogenous nitrogen level in the field. In 2020, Proven was used on 250,000 acres. Farmers using Proven can increase yields while also eliminating 1 gigaton worth of carbon dioxide equivalent emissions. Moreover, it costs less than chemical fertilizers. Undoubtedly, it is proven to be the better nitrogen.